So hello friends, welcome to Medicine 101. Today we'll talk about apoptosis. Okay, so before going in the topic, I have already uploaded the video in irreversible cell injury where I did talk about the necrosis. Okay, so it's also on kind of the thing in terms of the irreversible cell injuries. Okay, so apoptosis is over here. That is basically what the programmed cell death. Okay, so the apoptosis, it may be physiological or it may be pathological, but in terms of the necrosis, always it will be what pathological. So that means abnormal over here. Okay, now there are certain types of the genes that can that can stimulate the process of apoptosis, and certain types of the genes that can inhibit the process of apoptosis, and there are also one type of the genes that can just do the balance in between apoptotic genes or pro-apoptotic genes or anti-apoptotic genes. Okay, so we'll see all these things in terms of the first in the genes. Okay, so what type of the genetic factors are over here. So another thing is over here that apoptosis always you will see it can be seen in a single cells or some group of the cells, but always the necrosis we will see in terms of the large group of cells, or you can say in terms of the tissue. So tissue death will be in terms of the necrosis. Now let's talk about it's the programmed cell death, or you can see over here it can affect the single cell. Okay, it can affect the single cells. So single cell, or you can see some group of cells. Okay, the next thing is over here, there's certain types of the genetic factors of the genes that can just stimulate the process of apoptosis. They are known as the pro-apoptotic genes. So pro-apoptotic gene. Okay, for example, the genes are just like this. Let me use another color for it. So these genes will what? They will stimulate the process of apoptosis over. They are what? The BAC gene or BACs, or we can say the P53 genes over here, and certain types of the drugs that can also stimulate the process of what? Apoptosis. For example, the glucocorticoids. Glucocorticoids. Okay, these genes. And another types of the genes in recent exam it came that, for example, that is what? Basal. Okay. X. So these genes can also stimulate the process of apoptosis. And there are certain types of the genes that can what? Inhibit the process of apoptosis. They are known as anti-apoptotic genes. So anti-apoptotic genes. Okay, that means they will what? They will inhibit the process of apoptosis. And whatever the genes will get over here, the first and most important one over here, the BCL2 over here, you can get it. Okay, you can get the BCL2. You can get. Okay, let me. BCL will get. XL will get over here. Okay, and certain types of the drugs you can also get it over here. For example, the sex steroids. Sex steroids. So this type of the drugs can also stimulate the, they can also inhibit the process of apoptosis over here, steroids. Okay. And let me use MCL1. These genes. Now see, out of them, this BCL2 genes, okay, this is the most important one, which quite, the questions can be asked from these genes. Okay, so if you see in the later video, in the subsequent parts of the general pathology, in the general pathology we'll talk about these things. Okay, that can cause the lymphoma over here, particular lymphoma. And this gene, the MCL1, this is the most important in case of what? They just do as our resistance. Okay, they act as the resistance in case of chemotherapy. So chemotherapy induced apoptosis in these cases. Where? In chemotherapy. Okay, and there are certain types of the genes that will just do the balance in between the pro-apoptotic and the anti-apoptotic genes. Pro-apoptotic and anti-apoptotic genes, these are known as the sensors. So the sensors, they are known as BH3 genes also. And what's the main functions? They just do the balance, or okay, in between. Let me write it over here. Balance in between pro-apoptotic and anti-apoptotic genes. Pro-apoptotic and anti-apoptotic genes. Okay, this is the thing over here. And whatever the genes will get in this cases, like in terms of the sensors over here, the R. Okay. Sensors. We will get the genes like bead. Get it over here. The bead. We can get the beam. We can get the puma. We can get also the noxa. This type of the genes we can get over here. Okay. Now we will talk about how the apoptosis process is going through this all types of the genetical factors over here. Okay. Now for that thing, I need to draw a mitochondria over here. So for that thing, let me draw it over here. Okay. Now see, for example, is the cell. Within the cell is the mitochondria over here. Okay. And where we know that there is the presence of what? Cytochrome C. So the cytochrome C is present over here. Okay, this is cytochrome C. What is the main function of the cytochrome C? They help in oxidative phosphorylation. So oxidative phosphorylation. Now think about one thing. If this cytochrome C is not present within the mitochondria, then whatever thing we'll see over here, the problem will be here. The mitochondria will be not able to produce the energy. So as a result, we'll see the death of the cells. That means apoptosis. Now there is presence of certain types of the channels or because of proteins over here, genes. Okay, for example, the BCL2. Now this BCL2 always inhibit the cytochrome C to go outside. That means they will not let the cytochrome C. That means the BCL2 gene over here, we can say that will not let the cytochrome C to go outside from the cells to outside. Okay, from the mitochondria to outside. Now these BCL2 genes, okay, that depends on certain types of the growth factors. Certain types of the growth factors that can regulate the BCL2 genes. Okay, so that can stimulate this one. For example, if we see for any regions, if the growth factors, if you see the deficiency of the growth factors over here, okay, if we see the deficiency of the growth factors, as a result, the BCL2 genes, these things will not work properly. Rather, whenever the deficiency will be over here, okay, we will see the back gene, these genes, they will be activated over here. Whenever the deficiency of the growth factors, these genes will be activated, and they will allow the cytochrome C to go outside. They will allow the cytochrome C to go outside from these things. Whenever the cytochrome C is going from the inside to outside, as a result, we will see there will no oxidative phosphorylation. As a result, there will be no production of the ATP. So ATP that will be not produced. As a result, we will see what the cell death. Okay. So to maintain the growth factors over here, we need to have the enough growth factors to regulate the BCL2. Then the BCL2 will not let the cytochrome C to go outside of the cells, okay, or outside of the mitochondria. Whenever the deficiency of the growth factors will be over here, there will be stimulations of the back genes over here, and they will allow the cytochrome C to go from inside to outside. As a result, will be oxidative phosphorylation. There will be no oxidative phosphorylations. If there are no oxidative phosphorylation, there will be no productions of the ATP. Okay. Now I'll talk about the in terms of the types of the apoptosis. That means the apoptosis can be physiological or it can be pathological. Okay, so if I say it over here, for example, I am writing it over here, that's the physiological apoptosis. Now if we see the physiological apoptosis, where can we see the physiological apoptosis? So it's the first thing in terms of the embryological aspects. Okay, let me use another thing. Let me use this one. So the physiological apoptosis we can see in the embryogenesis. embryogenesis, where for example we can talk about in terms of the hand development. Now think about one thing. Whenever during the embryogenesis, we know the hands. Okay, the first of all, initially the hands are looking like just like this. Now see, there is no gap in between the fingers. There is no gap in between these fingers. Okay. Now we know that whenever the embryogenesis is process will be there. Now in between the fingers, in between these two fingers, okay, there will be what, the apoptosis. The process of the apoptosis will be there. As a result, we see in between the two fingers, the cells, they will just do what, apoptosis. We will see the apoptosis in between the cells of the two fingers. Then we can stretch just like this. So we can walk in the better way. So it's the physiological what, apoptosis, embryogenesis in the hand development. The second thing we can talk about over here, that in the reproductive age in the female, okay, the menstrual cycle, okay, during the menstrual cycles, we know in the reproductive age, we know what, the breakdown of the endometriums. So this is also what, apoptosis over here. So it's also physiological apoptosis. So the what, the breakdown of endometrium. Okay. The third thing we can talk about in terms of, for example, in terms of infections. See, whenever we will see the infections over there, whenever we will see the infections, definitely during any type of the infections, the total leukocytes, okay, that total leukocyte count, that will be elevated. Normally we know the leukocyte count around 4,000 to 11,000. Okay, for example, during infection the total leukocyte count is going around, it's around in 14,000 or 15,000 over here. Okay, so we'll see after certain time, whenever the infections will just go, that will come under the healing process. We'll see certain, okay, there is what, there will be no that much amount of the total leukocyte count. That means the whatever the leukocyte that came over in terms of the infections, all of the leukocytes, they will just do the apoptosis and there will be cell death of them. Okay, so in these cases during infections we will see the total leukocyte count, that will be more, but at the time, whenever the healing process will be there, okay, the total leukocyte, that will be again, leukocyte count, that will be again normal over here. Now we can talk about in terms of in the selections of B cell and T cell. In selection of B cell and T cells, we know these things, that what, in the bone marrow there is what, maturation of the B cells, in the thymus there is maturation of the T cells. Now just to test these things, okay, in the bone marrows or in the thymus, we need to check these cells, okay, by doing the MEC process over here, the MEC, major histocompatibility, major histocompatibility. In these cases, or we can say ACL also, this one over here. In these cases, we will see if the cells, okay, they just attack the cell tissue, then what, they just go under the apoptosis process. And if they don't attack the cell antigens, they will not go under the process of apoptosis in these cases. Okay, so in terms of the selections of the B cell and T cells, we can see the physiological apoptosis. Now we'll talk about in terms of the pathological apoptosis over here. Okay, so let's talk about the pathological apoptosis. Now, where we will see the pathological apoptosis? Now see that pathological apoptosis, if we see that any type of the exposures or radiations on the chemical exposures or the radiation on the DNA, then the cells is supposed to do suicide at the time of the, so they will do the apoptosis. So we see, if we see the exposures of, or you can say the radiations or the chemical exposure on DNA, then we will see this type of the process over here. That means the pathological apoptosis in these cases. Then we can see in terms of the duct obstructions. Okay, let me show you this one. So duct obstructions, for example, is the duct, and within the, for example, within the what, in the salivary glands, in the saliva, or in the pancreas. In these cases we can see. Now see, in these cases we will see the cells that are present where within the ducts, they can just do the apoptosis over here. What is the duct over here? Okay. So duct obstructions. Then we can just we can see also the pathological, apoptosis in terms of some viral infections. So in terms of the viral infection, now see, in terms of the viral infections, when for example if we talk about in terms of the liver, okay, in these cases we will see some cells, okay, whenever we will see the under histopathological conditions, if we just see under the microscope, we can see some cells that can go with the apoptosis process, okay. So that can be decided by seeing the concealment bodies. So concealment bodies, okay, you can just do the diagnosis by seeing the concealment bodies over here, okay. Now we can also see the pathological, apoptosis wherein the tissue versus, the tissue, for example, if someone is having someone needs the tissue transplant, okay, the organ transplant in these cases, in some cases it needs. So at the time, for example, whenever we are using some organ from another person's, another individuals to the recipient, then the recipient's immune cells will think the foreign bodies for the whenever he is taking the organs or the tissues from the other individuals, then the immune cells of the recipient, they will think it as the foreign bodies. At the time they will just do what apoptosis of the particular organs, okay. So in case of the tissue, in better we can see, in the better what we can say what, okay, graft rejections, host versus graft. In these cases we can see, and we can also see in case of like the retinitis, okay, there's like the different thing in terms of the rod cell and cone cell, we can also see these things also over here, okay. So this was all about the pathological apoptosis, and this was all about the physiological apoptosis over here. Now in the subsequent parts of the videos we will talk about the different, there are two ways of the apoptosis, extrinsic way and the intrinsic way. In the extrinsic way we will talk about how these things are getting activated. Actually there are two ways to do the apoptotic process over here. One is extrinsic and another intrinsic. Intrinsic way will be done by the for the mitochondria from these portions, okay, and extrinsic for the cell membrane receptors over there. So we'll talk about those things in the subsequent parts. So thanks for watching for this video, and if you don't understand a point, please let me know in the comment sections. I'll try to help you guys there.